Thank you, Leigh, for joining us today. So Leigh is Associate Professor in the Department of Biomedical Engineering in the School of Chemical and Biomedical Engineering at the University of Melbourne, and Director of the Melbourne Brain Centre Imaging Unit in the Department of Medicine and Radiology. So she focuses, her background in NMR, magnetic resonance imaging, and she's focusing on medical imaging. She's also Assistant Dean, Research Training in the School of Engineering. And her interests really centre around developing new sequences and analysis methods for using high field MRI and applying it to better understand brain diseases and disorders. So I'll leave it there, and I'll hand over to Leigh to tell us about brain imaging. Thanks, Leigh.
Thank you very much, Maria, and thank you for the opportunity to come and talk to you all today. So I'm going to share my screen and see how we go here. So hopefully this is now viewable. Yes. Okay, great. So as Maria said, I am from the University of Melbourne, and I live mostly in biomedical engineering from a day-to-day lecturing kind of aspect of my job, and then all of my research is in the Melbourne Brain Centre Imaging Unit, which I have been running for a couple of years now. And we have, so to start off with, I should add, and this feels a little odd. I'm trying to get the people on my screen vaguely visible. I know most of you have your cameras off, but it's this lacking the interaction of a normal audience that feels a little strange. It's like lecturing to a class where you can't see anybody's faces. I put on shoes for this webinar. I put on shoes for the webinar and felt very, very formal about it. You know, I've got a good shirt on, so we're all set for a proper webinar here. So, okay, let me explain what our outfit is and what I'm going to be talking to you about today. So the Melbourne Brain Centre Imaging Unit, we are in a building which was called the Melbourne Brain Centre. For want of a better term, it's now named after some rich donor, but you get the idea. It's a neuroscience building. So at University of Melbourne, it's the main headquarters for the Florey and also the university's neuroscience research. And in that sense, we have two scanners that are both clinical scanners, human scanners. So we have both Siemens, a PET-CT and a 7T MRI. And what I'm going to be talking to you about today is some work that we do on the 7T MRI. We are a, reasonably sized group, I guess. We've grown a little bit since I put these figures together, but, there's about 8 of us on the staff side and 8 to 10 at any one time, so on the student side, and we cover a whole variety of different human imaging projects, research. We're also a core facility, and we belong to the National Imaging Facility, and what that means is that we don't just scan for ourselves, we scan for absolutely anyone that wishes to come and pay us money to do the scans. We provide a service as well, and so collaborate widely with people around Australia. And you might think that an MRI and a PET-CT, you can find them in every hospital, but our 7T machine is quite special because there's only 2 of them in Australia. There's one at the University of Melbourne and there's one up at UQ as well, and they were both purchased about 5, 6, 7 years ago as part of an EIF grant. And if you know much about NMR, which I suspect there is a bunch of people on this call that do know way more about NMR than I do, 7 Tesla doesn't sound particularly impressive, right? So it's 300 megahertz. But when you consider that it's a clinical scanner with a bore size that fits a human into it, that's where these field strengths do get impressive. And that's why 7T is kind of like cutting edge is probably 11.7T, and there's maybe 2 of those systems around the world. But 7T scanners, there's about probably 100 of them, maybe a bit more in existence around the world. So they're certainly not your stock standard scanners. And it allows us to do types of imaging that are just not feasible at lower field strengths. So I'm just going to go back a slide. in the title, I did have, imaging biometals in the human brain using high field MRI. And I've put in the ultra high field because I recently had this kind of debate with people, and apparently high field actually means 3 Tesla and above. So that's if you go into any hospital, any good hospital will have 3 Tesla scanners at the moment. And if they start, if you need to have your brain scanned and they say, just ask them what field strength they're putting you in and say that you demand a 3T these days, there's no excuse not to have it. Ultra high field is that next step above, and that's what we're at. And I must admit, with over 100 of them in the world, I start to think, can we really call ourselves ultra? But it is still the name that is given. So we're talking about ultra high field imaging here. So I'm going to talk about 2 different pieces of research that, areas that we work in. And the first one is about imaging iron in the human brain. And the second one is about imaging sodium. And the first one, we image iron using a technique called quantitative susceptibility mapping. And that is the QSM took MRI by, not surprise, but it was all the rage, in the last 5-10 years. And it's still not done particularly well for reasons which I'll hopefully articulate. And we, like everyone else, we have our own QSM method that we like to push, but we think ours is particularly good because we have the best acronym. It's an artifact reduction technique, so it's QSMART. And we're well on the way to QSM domination with that, I'm sure. The second thing, sodium. So again, for those of you that know your magnetic resonance, sodium has a different resonance frequency to standard proton MRI, and so it is a whole nother ball game. So I'll spend roughly equal time talking about each of those. So if we talk about QSM to start off with, when you look at an MRI image, I would say 99.9% of the time you are looking at a magnitude image. So that which comes off a scanner, I'll see if I can get my cursor working here properly, there we go. That which comes off a scanner is a magnitude image of signals that have been received in quadrature. Okay, what does that mean? It means they're 90 degrees out of phase. You've got a cos and a sine component. And so 1.5 Tesla scanners, 3 Tesla scanners, the magnitude was the only thing that actually made sense. Okay, it's the only thing that gave the beautiful anatomic information. When you get to 7T, however, the phase image, and literally just the phase of those complex numbers that are the signal that's being read off at every point in time, the phase images actually have detailed information in them. And what the phase image tells us about is literally about the off resonances. Okay, so how far beyond your bulk magnetization as it is returning to equilibrium, how fast or slow relative to that average is the phase within a particular voxel, a volume element that we're imaging within. What is it doing? And there we ask the question, well, if we have a non-zero phase, then what does it tell us about the underlying tissue? And in a human brain, there aren't that many things that can cause observable off resonances to the point that you get these phase differences. So I'm not talking about little NMR type peaks around the water peak, right? They are unobservable to us. We just don't have enough field strength for that. But when you start to think about iron and iron being present in the brain, that is enough of a resonance frequency change to our proton signals that we can actually detect that. So you look at this phase image down the bottom of this slide here, and you think that's not looking particularly impressive. Okay, there's not that much information in that. But hopefully what you can kind of see hinted at the back of it is that there's a cloud that overlies this image, and underneath is actually some really amazing information. So there is a direct relationship, as I was saying, between the phase images and the underlying resonances. And those off resonances are all determined for us by the magnetic susceptibility constant, the chi. So we talk about doing QSM mapping, quantitative susceptibility mapping, but what we're really trying to do is just find the best estimate we can of the magnetic susceptibility at each voxel. And at that point, if we're able to take our phase images and use our magnitude images in a smart way to come up with estimates of the magnetic susceptibility, then we say that we have imaged iron plus a whole bunch of other things, okay? So this is really, it's the kind of thing that you present these images to people studying neurodegeneration, and I have no idea if there are people on the call that, you know, fall into this camp, but you can certainly question me about it later. People get really excited about this as neuroscientists, this idea that you can image iron, but we have nothing that actually says, well, that is iron, this is ferritin of a particular type, and that voxel next to it isn't. All that we can say is that we have an estimate of what the magnetic susceptibility is. So I am very keen always to point out the limitations of the methods, and this is a limitation. I cannot actually say we are directly measuring iron, but there have been plenty of studies to show that there's definite correlation. For example, early studies showing histology images with plaques located and showing that they co-locate in terms of an MRI scan. Okay, so we've got a pretty good idea that that's what we're looking at. So let me talk a little bit about more about this. So with QSM, as I said, you're estimating these Chis as a spatial map from the phase data. It's not just the iron. Okay, so the iron being anything that's paramagnetic or ferromagnetic is going to lead to a positive susceptibility value, or contains a positive susceptibility value, which essentially pushes our phase information one direction around the average. On the other side of things is myelin, and that has a negative susceptibility, and myelin is what is wrapped around each of the axons in your brain. It's a lipid bilayer, and it's extremely important in diseases such as multiple sclerosis, because that is actually what degenerates. Sometimes it comes back, sometimes it doesn't, but it's like unwrapping a piece of copper wire and having all of the data just disperse into the environment rather than being picked up between the repeater stations that are neurons, if that makes sense. It slows down the conduction, and it means that the signals are not being transported efficiently around the brain if there's no myelin. So QSM, while people are excited about iron, people are also equally excited about the potential to distinguish where there is iron accumulation and where there is myelin degeneration. So coming to the physics of this a little bit more, if we have an off-resonance, some kind of a non-proton magnetic susceptibility, and I won't say non-zero, but something that is different to its environment, then it affects the environment around it by a standard dipole model. Right? So the signals that we end up measuring, it's a volume integral over space of this dipole kernel, which is convolved with this underlying susceptibility map. So this is the D convolved with chi. And we measure our signals, which are field shifts or the phase map, which is equivalent. If we know the timing of our imaging, we can go from our phase maps to our field distortions. We need to be able to get from the signals that we measure, from our knowledge of a dipole kernel, what this chi map actually is. Now, so as I said, we've got phase maps on one side, dipole kernels, susceptibility maps. To solve this in space is extremely time-consuming and difficult. And so, because convolutions aren't nice things to deal with, what would we prefer to deal with a convolution? Put it in the Fourier domain. Convolutions, of course, turn into multiplications. And so here, this picture down below is all over QSM literature. What that picture is representing is this Fourier space version of the dipole kernel. However, there is a problem with this, and it's this equation on the left-hand side, if you're interested, where there's this three-dimensional K space or Fourier spatial frequency space. When this value is equal to 0, then we have an unsolvable problem, essentially. Okay, so our inversion becomes ill-conditioned, and this causes problems and artifacts for us when we're trying to get at this chi value. And so all of these QSM methods that have been in the literature for so long, and the reason that there isn't just one way to do this is because everybody's trying to work out how to remove the artifact the best. Okay, so to give you an idea what these chi maps end up looking like and some of the problems that are there, this is a coronal section of somebody's brain, and this on the right hand side is a sagittal section, so cutting along the midline or near to the midline. I think even if you are not brain imaging experts, you would agree that the anatomical information here is being obscured by black and white clouds to lesser and greater degrees, and that there is artifact in most parts of this image. Okay, and we've just highlighted a few of them, such as this dark banding around here is definitely caused by the sinuses at the base of the brain around your nose. And remember that air, or not remember, air has a very different susceptibility to brain tissue, okay? And so that itself is causing these susceptibility changes up into the brain, and we have to take all of that into account as well when we're attempting to get down to the chi values, to the iron and the myelin correlates. So this is the kind of images that we started off with that we wanted to try and improve. So it would, as I said, every person and their dog is out there trying to find the best QSM method. The one in the middle here, and so this is from a review paper taken from 5-6 years ago. The ones ringing around the outside are 8 different QSM algorithms. So they're all imaged the same. You just push a different button at the end, you'll get a different answer. Right. The one in the middle is known as the gold standard, and it's called Cosmos. I can't remember why it's called Cosmos, but everything in MRI has an acronym associated with it, right? Because that's how you get people to remember your method. So what Cosmos does, and again, if anyone ever asks you to be a volunteer for an MRI scan for a study, and you say, well, are you doing Cosmos? If they say yes, you might think twice about doing it. Because what Cosmos does is try to, you don't just get imaged once in a comfortable head position, but your head is tilted to the side at different angles. So what Cosmos is attempting to do is see the brain structures at differing angles to the main magnetic field. Okay, because there are all sorts of anisotropies in here, which I haven't talked about, but essentially imagine this dipole kernel. If you can see the tissue at various angles to that dipole kernel, which is defined by the main magnetic field, you've got a better chance of undoing it, right? And that indeed you can see that this image, well, I'm not sure actually you can see that, but this one in the middle is actually better than its surrounds. And I should point out, sorry, this one down on the bottom right-hand side is the phase image from which the susceptibility values estimates are taken. Okay, so we have something like a gold standard to work with, but it takes, you need something like 4, 5, 6 different head orientations. It's extremely uncomfortable to hold your head in an angle to, for 5 minutes at a time to be able to take a full 3-dimensional anatomic scan. And in a clinical setting, certainly nobody has that kind of time to waste, particularly when you're up against a CT machine, right? And the CT does things like this, and the criticism of MRI is always it takes so long, all right? So you're not going to get anywhere with clinicians if you tell them that they have to take 5 times as much data to get there. All right, so what we've done with QSmart is firstly, we've taken a brain mask from the magnitude image, which is very simple to do. Right? This is standard, okay? But we know that around the edges of the brain, we're getting a lot of information kind of folding over from the high susceptibilities or the high relative susceptibilities outside. We also know that it's the vasculature within the brain that is causing us issues as well. So what we've said is, well, you need to take account of these you need to take account of what the vasculature, and this is high susceptibility because there is iron in the blood. All right, so there's off resonances all through there. So the first part of our method was let's clear up stuff at the cortical surface. We need a mask of that. Let's clear up things in the vasculature mask. And we also created what we called an indentation mask to deal with the fact that everybody's sinuses look a bit different, etc. Okay, so this is all done on the individual subject. So then what we did was say, well, what we want to do is to remove this kind of background field that lies on top of the phase image that we have to get rid of to expose the details. We want to do that by varying the filter sizes that we look at. And so we do something called spatially dependent filtering based on the cortical surface mask, based on the indentation mask, and based on the vasculature mask. And I won't go into details of that now, but of course, if anyone actually works in this area and is interested in it, I'm more than happy to chat at any point about this or answer questions. Okay, so we clean up essentially our phase images to get nice, what we call local field shifts. We've removed these clouds and we're exposing the details underneath. And we get rid of the high susceptibility sources that way, and we do an inversion to find the chi map. But at the same time, we do a whole brain inversion. And then at the same time, we do a tissue only where we've actually removed the vasculature from from the whole brain. You can see the difference between removing the vasculature and keeping it in. You get quite some startling, high susceptibility artifacts lacking there. So we then combine these two things into our finished image, which is our QSmart image. Now you look at that and you go, oh, well, it looks all right, I guess, don't really know. Well, let's look at it in comparison to competing methods. All right, so in our method, QSmart is at the top, and again, it's sagittal and coronal, just switched from what I had them before. And in the middle, this is like the state-of-the-art best method, ReSharp ILSQR. Don't worry what that stands for. It's just somebody else's QSM processing method. So the differences, I think, are fairly obvious, and we really haven't cherry-picked here. It is literally, if you do these masking things properly and take out the artifacts, take out the artifacts by a principled physics-based modelling method, it actually makes sense, and it really does work. And you can see that in the difference images below. And if you'll also note, in the difference images, I think you'll see something of a cross, kind of a streaking cross-shaped artifact. That's all due to that, the shape of the dipole kernel in the Fourier space. And so we know that if we're cleaning up our images, we're getting rid of some of that, that dipole-shaped artifact, which is good. So here's just some more examples. This is the axial section, so going flat across the brain, and some zoomed in images, which just visually show us that, yes, QSmart actually works, and we can be hopeful that the susceptibility values that we've got actually can be meaningfully analysed in terms of potential source of iron or myelin. We did do some quantitation there, because quantitation is always a nice thing. I wouldn't worry about this diagram, except to say that we analysed the variance of our estimates of the susceptibility as we got further and further away from the brain edge or the vasculature mask. And it all makes sense that as you're further away from these high susceptibility sources, methods come together. But ours, the red line, is working much better as you're closer to these high susceptibilities. So, we're happy with that. So just in summary about this, QSmart is working really well compared to other methods. We've tested it at 7T. We've tested it also at 3T, where the phase information isn't as good. And you think, well, what's the relationship there? So remember that you've got a resonance frequency, which is telling us how fast our signals are processing, okay? What's its angular frequency? At 3T, if it's processing slower, if the protons are processing slower and the bulk magnetization is oscillating slower, so your B1 fields are slower, your off-resonance differences aren't as pronounced. And if they're not as pronounced, they just don't look as good. You can't get as much information out of the signal. So that's why it's really great to be able to work on this at 7T. On the other side of things, we've also tested this on 4.7T and 9.4T animal scanners, and it works really nicely. So we're convinced that we have a nice method here. It's being used by people at various groups across Melbourne and Monash. And as I said, I'm sure it's only a step away from QSM world domination, but we shall see how that goes. Our first step is actually getting the reviewers' comments back, which are due next week. So, all right, now, so that's iron. And as I say, it really is exciting to people, particularly neuroscientists who study neurodegeneration and brain diseases, to consider that they might actually get these kind of, but these quantitative maps. I, however, will go back, and I'll just put up this slide again. I've deliberately tried to show you a bit of the nitty gritty of what it takes to go from the measured signal into these maps, right? There are so many tuning parameters in this, in terms of filter sizes, number of iterations that you do something for. I think when we claim that we're estimating magnetic susceptibility, we need to keep this, we need to have this, a realistic picture of it, to say we won't immediately go and search for something that's at 3.5 ppm and say that that is a particular type of molecule. Okay, that's really not what we're trying to do with this. However, it is clear that the susceptibility maps really do track stages of neurodegeneration, particularly in Alzheimer's disease. So people are excited by this. Okay, I will keep moving. to the second thing that I wanted to tell you about, which was the sodium imaging that we're doing. So what I'm going to be talking about is our attempt to put sodium and proton imaging together. So in a multiplexed way, which one of my collaborators, my postdoc, has called MQ'd-N, which I don't think actually rolls off the tongue. You can give me your impression of that, but we're still looking for a better name, but multiplex measurement of multiple nuclei. So here's an introduction to sodium. Let's say that ultra high field is very necessary. And you might think that if I am writing this, then what this one at the top is sodium images taken at 7 Tesla, and down the bottom, sodium images taken at 1.5 Tesla. But unfortunately, that's not the case. At the top here, these images are 7 Tesla images of proton channels, so water, okay? Down the bottom, this is what sodium images look like at 7T. All right, now sodium has 1/4 of the resonance frequency of hydrogen. So rather than working at nearly 300 megahertz, when we're imaging sodium, we're down at around, I think, 78, don't quote me on that, yeah, 78 megahertz. That then means that puts us back in the realm of the 1.5T scanners. And this is kind of what brain images used to look like, taken 15, 20 years ago on a 1.5T scanner. So we're in the right realm, but of course, this is why I say high field is very necessary, because if you're doing this with anything less than a 7T scanner, you're at 1/4 of the resonance frequency, you're just not going to be seeing anything useful. Okay. So why do we persist doing sodium MRI if this is what our images look like? Well, the fact is that sodium is, I don't think I would have to convince anyone that for biology, sodium, and particularly human biology, sodium is a very, very important quantity, all right? So whereas the proton information gives you information, the images give you precise anatomical information, sodium is much more, well, not much more, but it is linked to physiological processes in a complementary and distinct way to what the water, the hydrogen is. So the hope is not that we're going to repeat not that we're going to get the absolute beautiful anatomic information. What we're looking at in here is information about cell integrity, cell viability, for example. We want to know about differences between intracellular sodium versus extracellular sodium. All right, the point being though, that usually anything X nuclei, and to explain what does X nuclei mean, it means anything that's not proton, all right? So sodium falls into X nuclei, and you need, when you, so I should tell you that we have a sodium coil that we paid, I don't know, $500,000 for or something like that. These are expensive coils. I don't think it was quite that much. Oh no, I think it was 400K or so. The other 7T scanner in Australia, up in Queensland, made different choices with what kind of areas they were going into. They don't do sodium imaging. So one of the 3T scanners across the road from us at the Royal Melbourne Hospital has a 3T coil, at 3T has a sodium coil associated with it, which, you know, has dubious quality. But there are very few sodium imaging for humans around, okay? So it's quite a niche area. And again, we have the problem that if you tell a clinician, well, you can get these kind of dodgy images that look something like these ones at the top right, and it'll cost you an extra 20 minutes. They'll just go, forget it, not interested. All right, so what we wanted to do was see if we could put the two things together. So we could use unused time in scans that we do routinely for the proton and kind of slip in the sodium scan. So let me tell you about this, okay? All right, so this is this idea, this multiplex measurement of multiple nuclei. Now here we have combined two sequences. I'm getting confused by multiple screens, but here we go. Marina, well, I'll explain it on the next page actually. Okay, so we've got MP2 RAGE, which is a stock standard proton scan that we do. Every person that comes in gets a scan, no matter whether they're doing functional, diffusion, whatever. We do a beautiful T1 weighted anatomical scan. That's called an MP2 RAGE. I apologise if things are getting noisy. My children came home from school and are yelling outside the door. So I'm hoping that at some point, if I mute and yell, that's what's happening. Anyway, so MP2 RAGE. What this does is it uses, and again, I'm not sure how much detail to go into here because NMR people, this will make sense. Other people, I'm sorry, it may not. We invert. There is a couple of inversions in an MP2 RAGE, which means that you start off by flipping your magnetisation down to 180 degrees, and then you wait a period of time before you do the actual imaging. All right. And that's what it means down here when I say magnetisation prepared, gradient record echo readout. Okay, so whenever you invert and then you wait, it means that there's a bunch of time that's not being used. Okay, so that was our first kind of what can we do with this? On the sodium side of things, the Marina sequence is one that we developed a couple of years ago, and it's a multi-echo sodium, a radial sequence. So sodium disappears so quickly that as soon as you flip it to 90 degrees, you essentially have to start imaging before the signal disappears, or else you can't build up a good image. And so what we do, though, is we do go back and forth through the centre of K-space to attempt to build up some multi-echo information as the signal is getting smaller and smaller so that we can actually do some parameter mapping. Okay, so it's a standard 3D radial multi-echo sequence. We want to put these two things together. So here's the idea. An MP2 RAGE essentially falls into these blocks, and we talk about TR1s up to TRN. TR means the repetition time. So in an MRI scan, you're doing it every time you're changing slice. So there are blocks. So here's our imaging. Every time there's a solid block here is when you're actually doing the imaging, when you're actually reading out information. Where there is empty space is where you're waiting for the magnetisation to get itself into a particular contrast that you want to see. So if we put the sodium scanning, the Marina, which you can see is very, very dense, okay? We're not losing any efficiency here because we want to get the information as much as we can, as quickly as we can. We want to take that and instead interleave it into the MP2 RAGE, because that then means that we get two for the price of one. So we've got a fixed total acquisition time, and we're filling up that scan with the sodium. The scan one, get one free. That was our principle. All right. So I show this not for any particular reason, but to say you'd think that this would be easy, right? So we have these concepts, right? And we go, oh, that's neat. All right. Has anyone else tried this? Okay. So over the last 30 years, yes, people have talked a little bit. There's a few papers around about trying to do proton and another nucleus at the same time. So we talked to our good friends at Siemens, and we have a really nice, close working relationship with Siemens. And we say, okay, so we want to apply two different excitation frequencies in the same scan. All right, so we want a B1 field that's turned on at 300 megahertz, and we want to then closely interleave it with something that's turned on at 78 megahertz. And they're kind of like, well, actually, you can't do that until you get our next upgrade. It's kind of like Windows. No, I shouldn't say that. So we are actually, in the process of upgrading the scanner to make it, the next generation, where they have actually allowed this to be possible. But at the moment, we had to go and get this little device from a group of people in Germany who had also attempted to do this. And essentially, it's plugging in a bunch of things around the back of the scanner that you hope to never have to change. But what it does is it allows us to do this multiplexing so that we can actually read out, both excite on two different channels and read out on two different channels. And I will not go into the details here, but that was the details of how it was done. Okay. And then, of course, nothing works as you would expect it. And there's a bunch of post-processing that you need. Well, not post-processing. So in the image, inside the scanner, you've got the sequence that plays out the magnetization, the B1 applications, and then you've got the image reconstruction. What we're talking about is in between those two things, there's some adjustments that have to be done to get it all working happily. Okay, so this is what our sequence end up looking like, is that we've got these 90-degree flips in here on the sodium channel. And of course, the 180s here are on the proton channel. So they're not actually affecting the sodium signal because they're at 300 megahertz rather than 78 megahertz. So this all works really nicely. to interleave the two different sequences to do the reading out and all the rest of it. But for those of you who are attuned to the ways of magnetic resonance, and particularly when you're trying to scan a human, you might be thinking, well, doesn't this create power problems? All right, so we cannot fry people when they come and have their brain or any other body part scanned. And there are very, very specific SAR limitations, so specific absorption rate limitations on how much energy you can deposit into the biological tissue. And so certainly with this, we are pushing the envelope of that, because every time you excite up on this top line, every time we flip the magnetization, we're applying an oscillating magnetic field, which is depositing power into the tissue. So we do have to be careful with that. And essentially what that means is, rather than achieving 100%, scan one, get one free, in the end, we have to stretch everything out by about 10% to be able to do the imaging, but it really isn't a significant change in time. Yeah, okay. So we tested this out with our 7T and our dual-tuned sodium proton head coil that I was talking about. We did both phantom imaging, so we, phantom just being a geometrically known object that gets put in the scanner. We varied both agar and sodium concentration, and we tried it out on this at the moment in vivo on one of our group members. That's the fun thing about being in a brain imaging lab. You get to be guinea pigs for each other's scans. All right, let's not worry about those parameters, but they're all the various parameters that needed to set the scanning up. So here are some images from the proton side of things. And I have people's heads in the way here. I'll just move the images, your heads, so I can see these phantoms. So the top line is the anatomical images, quote unquote anatomical images of these vials in a bigger container that we take just if we were doing just proton imaging, and here is the combined, and there is no noticeable decrease in the image quality. And I've got to hit this again to be able, there we go. And we analysed that a variety of ways to show that that was all good. And then on the sodium side of things, and again, this is a multi-echo acquisition. So this is the strongest, and then we're coming back through the middle of K-space to be able to build up our echo information. No appreciable difference between doing it by itself or part of this dual, of this multiplexed imaging system, as one would expect. And, we also analysed the decay curves from it, and it all looked fine. So moving to the actual human brain images on the left-hand side. So again, it hurts my heart every time I look at this, because the proton just works so well, and the sodium is like the poor cousin, which as much as we would like, we're not getting the same anatomical information. But what we are getting out of this, and this is why we're, collaborating with people now that are studying schizophrenia, that are studying multiple sclerosis, a bunch of different brain disorders and diseases, because the sodium information, it may not have to be the best. It can look a little bit more like a functional MRI scan, for example, but it's about those relative differences between groups of people. So on the left-hand side, here's what's coming out of the MP2 RAGE sequence, and you can always form a T1 map from that. And on the sodium side, these are the multiple echoes that we are getting for the same person scanned at the same time. All right, so just to summarise, no increase in scan time. So we're very hopeful that this is going to be a type of technique that appeals to our clinical friends. Our images are registered implicitly, and this, again, if you don't work with animals or humans, if you don't have anything that moves, this may not actually be such an issue, right? But as soon as you put a living, breathing object in a scanner, every time you image them, even if they don't think that they've moved very much, part of a millimetre will make a difference, right? And so we're always going through this process of, well, here's what we scan now, and here 3 seconds later for this functional MRI scan, we've got to register these two brains together to make sure that they're in the same space. This is also a great advantage of what we've done here, is that our images are registered together. So even if you take them in different resolutions, they overlay each other very, very nicely. And hopefully I've convinced you that we get a rich multi-parametric imaging space to explore, essentially, here. And you can extend this to other nuclei. I personally don't think we're going to be investigating beyond sodium, but I know that Siemens are particularly interested in this. They're making a push for their 7T scanners in the X nuclei space. So they're working closely with us on this because they're certainly interested in this technology. Okay, so acknowledgements, Nagin and Werder and a bunch of other people here, Scott, etc., were very much involved in the QSmart, and then Yasmin has been PhD student and now an independent researcher working on the sodium, and our Qingchuan is our PhD student currently on the sodium. And this is a very old picture of what the brain center imaging unit used to look like, and of course this is what we look like now that we're all Zooming each other instead. So I am going to leave it there, and I'm very, very happy to take questions.
Very nice, thank you, Leigh. I'm sure there'll be plenty of questions. I might just start the ball rolling with a couple if that's okay.
Do you want me to keep sharing my screen, Maria? Or do I?
Whatever you like, up to you, I don't really mind.
I'll leave it just
leave it in case you want to ask questions specifically about your. So two questions. First question for me is the resolution that you can get in this sort of imaging. What's the voxel size? What sort of size are you able to look at?
Yep. So with the iron, with the QSM images, with standard proton images, we would typically be imaging around 750 microns isotropic. Okay, so this isn't fat slices. If we went to fat slices, then we could certainly push that resolution in plane, because of course it's all about the density of protons that you have. And so we like to keep things isotropic because it just means you can slice and dice it any which way and it'll look the same. But that's typically the resolution that we would use. If you did this at 3T, you're up at 1 mil cubed or more. But that's really where a 7T has some advantages. Yeah. With the sodium, however, you're back up at 2 or 3 mil cubed. Okay. Just because you take these radial projections and they're cush balls, right? They're, and the more spokes you take, the longer it takes, but the better the resolution will be when you grid them to a Cartesian grid. So
just on that, I think Gordon's asking, excellent, thank you, Leigh. Does the combined sodium and proton imaging sacrifice some of that resolution for both? Or
no, not at all. No, we can do it at exactly the same resolution for each. Yeah.
Yeah. Before I go to Jenny's question, with the sodium, are you imaging nuclei density? Are you imaging relaxation times? Are you imaging, what are you imaging? What's the actual measurement? I mean, not the measurement, what are you imaging? Relaxation times or density or?
So if we're able to image before the FID disappears, right? So as close to the density, we call that, and in the literature people call it the total sodium concentration.
Okay.
So that just means everything that's sodium that we don't actually know where it comes from. Thereafter, in those multiple echoes, that's where you can see the bi-exponential decay. So there's a fast component, a slow component, and we've got a variety of methods to try and pull them apart. So the multi-echo data is great in that sense. It's certainly not everything that you would want. So there's some other work that I didn't present today where we're trying, I don't know if you've heard of flare imaging, but it's essentially putting a 180 inversion on the front of a standard imaging, not like MP2 range, but and it nulls the fluid signal in a standard image so that everything else, you'd think that could just be done by image processing, but it really can't. It nulls the fluid signal that you get from the CSF, for example, the cerebral spinal fluid, and the contrast in the tissues is much better with a flare image. We're trying to develop the same thing for sodium, which is a bit tricky because the amount of power that you need to invert already starts playing with our resolution and stuff. But if we can do that, then we can look at more closely these intra versus extracellular components. Yeah.
Cool. Jenny says, can you use this technique for other areas of the body, like to look at degeneration in the spine or the joints? And
yeah, absolutely. The other area that uses sodium imaging most is cartilage imaging in the knee. So We have a knee coil, but we don't have a knee sodium coil. Our brain coil, and this is where at 3T there's far more coils that are available. And you can get away with using the big volume coil, which is actually kind of built inside the scanner. It's not something you add in. At 7T, for a variety of different reasons, you really need to be dealing with a specific coil for a specific body part. And so we've got the brain coil that we use for proton imaging. We've got this dual tuned one for sodium imaging. We've got these really neat eye coils that kind of sit on the face, and a knee coil, but the only one that we have that does sodium on is the brain one.
Right.
So yeah, there's a whole lot of literature around cartilage degeneration. I haven't seen anyone with a spinal coil specifically for sodium, but there's no reason why you couldn't develop it. It's just expensive.
If you imagine some of the stuff that Mark Cook and others and Gordon's team have been doing on the brain on the bench type stuff, so you haven't got a human body, you've just got an inanimate sort of static thing, bunch of cells. Can you look at, and can you get information from this, look at imaging that sort of artificial brain, if you like, and start to, because that must be much easier. A, you've got a high field, you don't have to worry about the human body, and B, you can probably do a lot longer experiments because you don't have to worry about.
Yeah, that's a great question. So the piece of the puzzle that I haven't mentioned, and I've mentioned David Wright, who's I think actually some of you may know him, but he was at the Florian and has been at Monash the last couple of years, and he is what I would describe as Australia's leading animal imaging person. Like, he really knows his way around a preclinical imaging system like nobody else. And at the Monash, and when I say Monash, I mean Monash Alfred, not Monash Clayton.
Yeah.
At the Alfred, they have this beautiful 9.4 Tesla animal scanner, and they have a sodium coil that they bought. And again, that's the only one I'm aware of in Australia. David and I have been saying, like, I've listed him here because he's been involved in the QSM work, but we have been saying since that scanner came on board that we were going to do some sodium imaging using that coil, and it hasn't happened yet. But that actually is an excellent suggestion, that these brain organoids, that would be the ideal environment, because then you're not dealing with the massive, like our gradients on a 7T are 70 millitesla per meter max. On a 9.4 Tesla, there's 700 or nearly 700 millitesla per meter. It just blows it out of the water. So yes, it's a great idea.
And Alex Harris says they're already having this discussion with David. My other question relating to that is, if you were to look at implanting electrodes into this, what would be the impact of that on image? I know we do, like, we try and do batteries and other things, and when you put metals into a magnet, ugh. I don't know that, but from your perspective, do you think it's a possibility to be able to actually do the organoids, whatever they're called, with the electrodes in there, and actually do in situ, in whatever?
Yeah, absolutely.
Cool.
I mean, firstly, of course, the issue is, can you find some non-ferromagnetic electrodes that aren't going to stick to the magnet and cause all sorts of problems?
Platinum.
Yeah, okay. So the issue, of course, then is about getting whatever it is that you're circulating. Presumably the pumps or whatever have to sit outside the magnet room. Yeah. So they need to have 2-3 meters of cabling. So long as that, there's waveguides that they can go out through, it is possible. It just gets a little bit
we do that with batteries. So we have cables coming out to a potentiostat, right, or through a thermocell. So if you're able to do that with this sort of imaging, that could be something to talk about with Gordon and Mark and others. It's actually really interesting.
Yep. Yeah, and I mean, from the human scanner point of view, it's not, of course, it's not that all metals are bad and all metals are, we routinely scan people with hip implants and all the rest of it. It's just about making sure that it's safe. And
safe, and also more from the point of view of resolution and artifacts again. Because every time you're near a metal, susceptibility artifacts create a problem. So you exactly get image close to the metal. So yeah.
So when we image people with braces themselves won't harm them. It's not like they're going to make them fry. But oh my goodness, yes, the big susceptibility artifacts you get up into the brain make it nearly not worth doing.
Okay, that's interesting. Any other questions? I think for me, it's fascinating. But any other questions from the other people on the call, on the Zoom link? No?
Maybe I'll...
No, that's it. All right, well, maybe I think that was fascinating from my perspective, Lee, and we'll have more conversations as we start to discuss sort of centres of excellence and whatever else. But Gordon, do you have anything else you wanted to say?
I just thank you very much, Lee, and yeah, that was great, and lots of exciting things to explore together, I think.
Yeah, very good. Anytime.
So from my perspective, using your knowledge with the body into devices, I mean, we look at sodium batteries, right? It's in electrolytes, so being able to image sodium would be pretty cool. So, all right, well, if not, let's thanks again, Lee, for agreeing to do this. I've learned a lot. Thanks you, and thanks everyone for coming along and listening.
Okay.
Now, bye.
Lee.